Hi everyone, welcome to the third and final lecture of chapter 11 where we're looking at nervous tissue and the fundamentals of nervous activity. We're going to wrap up this chapter today and then cover muscles next, but we will be coming back to the nervous system after that, and then the nervous system will take us through the end of the semester. So, as always, let's go ahead and get started with our not really attendance questions. First, where are proteins that are destined to leave the cell made? Next, how are those proteins transported around the cell? And third, when a cell becomes less negative on the inside of the membrane compared to the outside of the membrane, what's that called? Go ahead and pause the video, try to answer those questions. They will be important today. Okay, hopefully everyone has done that, so let's take a look to see what the answers are. First, where are proteins that are destined to leave the cell made? This takes us back to chapter 3 when we were studying the cell to begin with. Remember, proteins are always made on ribosomes, regardless of where those proteins are going to be used. Proteins that are going to be used inside the cell are made just on free-floating ribosomes that are loose in the cytoplasm. But proteins that are destined to leave the cell or become a part of the plasma membrane are made on the rough endoplasmic reticulum. It's still ribosomes that are making them, but those ribosomes are attached to the rough ER. Next, how are those proteins transported around the cell? Well, after the proteins are made, remember they're packaged in a vesicle, and that vesicle then travels and merges with the plasma membrane, and then whatever is inside that vesicle, that protein that was just made, is exocytosed. It's moved outside the cell. Third, when a cell becomes less negative on the inside of the membrane compared to the outside of the membrane, what's that called? We talked about this a little bit in chapter 3 and previously in this chapter. That is called depolarization. So cells are polarized, their membranes are polarized, because cells on the inside are negative compared to the outside when we measure voltage across that membrane. If they become less negative, if they're becoming more positive or closer to positive, that is called depolarization. If it were to go the other way, if it were to become more negative, that's hyperpolarization. Okay, so real quick, just a refresher of something we talked about in the previous lecture, or maybe even the first lecture of this chapter. Remember ligand-gated channels, also called chemically gated channels. Normally, they are closed and nothing can pass through. But if a ligand, which is a specific chemical, usually a protein, but not always, if the ligand binds to that ligand-gated channel, it's like putting a key in a locked door. It opens the channel, and then whatever substance that channel is specific for, in this case, it looks like it's probably sodium, but it could be a few other things. Well, now it's open, so that substance is free to diffuse through. And this doesn't take energy. This is just something binding to a protein. Remember, anytime something attaches to a protein or is removed from a protein, that protein will change shape. Here, that's being demonstrated. The ligand binds to the ligand-gated channel. It changes shape to open up, and then something can flow through from high concentration towards low concentration. Passive diffusion uses no ATP. So last class, we talked about action potentials. We had our plasma membrane, we had high sodium outside the cell, low sodium inside. We had high potassium inside the cell, low sodium outside. We had various different types of proteins. We had those leaky sodium channels. Sodium was always leaking in. We had the leaky potassium channels. Potassium was always leaking out. We had the sodium-potassium pump that used ATP to take 3 sodiums, move them back out of the cell, 2 potassiums, move them back into the cell. At rest, that was always happening. Well, regardless of what's going on, that's always happening. At rest, the cell was 70 millivolts, but if something caused that negative 70 to start to rise, if we had a bit of depolarization, if that 70, negative 70, became as high as negative 55 millivolts, we called that threshold, and that caused the voltage-gated sodium channel to open, and now sodium can come in. That negative 55 rose very quickly, as high as positive 30. The inactivation gate slams shut, sodium stops coming in, but the voltage-gated potassium channel opens. Potassium starts to rush out, bringing that positive 30 back down to negative 70. At negative 70, the voltage-gated potassium channel begins to close, but it's slow, so we overshoot. We get down to something around negative 90 before it finishes closing. So really what we had was a very brief reversal of charge in a very localized area. But that very brief localized charge had such a magnitude, it got up to positive 30, and it always goes to positive 30. It will affect the next area in line where all of the exact same proteins are there. So everything that happened here now happens here. And again, off screen. So it always goes from the soma end towards the axon terminal end. Something happens up here at the soma, and it causes the action potential to travel the length of the axon until we get to the axon terminal. All of that was in the previous lecture, so that was just a review. That's why it was kind of messy on the screen here. But remember the way that axons looked. We have the soma up on one end, we have the axon hillock, we travel down the axon, and then we had the axon terminals. And then we have another axon in line. But right here, we've got an open space. That open space is called a synapse. And the trouble is, this open space, you can't have proteins on a membrane if there is open space there. So how does the action potential travel across this open space to possibly cause another action potential? How do we pass the action potential along that open space at the synapse? That's what we're going to look at today. So we're going to draw. And as you draw this, I'm going to kind of talk through what it is that we're looking at. Over on the left is an axon terminal. We see the axon. Let me see if I have a laser pointer here. I do not. Well, over on the left, that's an axon. And that kind of bubbly looking area right here is the axon terminal. Remember, we called that a terminal bouton. I've drawn some sodium outside the cell. I've drawn some potassium inside the cell. But then down here at the axon terminal, there are some vesicles. That's what those black circles are. They are vesicles. And inside is a protein. Specifically, these are neurotransmitters. In this case, and every book that I've ever seen that draws this as an example uses the same neurotransmitter in the example. It is something called acetylcholine, abbreviated ACH. Usually it's drawn as little green balls. So acetylcholine is the neurotransmitter that we're using in our example. Now, we also said that calcium is high outside the cells, low to non-existent inside the cells. And if calcium is inside the cell, it's stored inside the smooth endoplasmic reticulum because it likes to bind to things, we said, and we don't want it to do that. Well, then on the other side, this is another neuron. And this area right in here, we're going to call it a dendrite. Remember, dendrites are the receiving parts of the cell. This could also be a soma, a cell body. It would behave exactly the same. I'm just going to refer to it as a dendrite. Well, over on the left is the sending neuron. Over on the right is the receiving neuron. We have the whole thing that we've drawn here is a synapse. The point where two neurons connect is a synapse. The one on the left, that's before the synapse, is called the presynaptic neuron. The presynaptic neuron is the sender. Over on the right is the postsynaptic neuron. The postsynaptic neuron is the receiver. The entire thing is the synapse. The space between is called the synaptic cleft. The synaptic cleft is the open space. Now, everything that we talked about in the previous lecture, the action potential that is being sent down the axon, we say the action potential that is propagating down the axon, is happening in this direction. All of those proteins that we drew are along this membrane, and all of those events that we drew are happening. Now, when we get to the end, when we get to the axon terminal bouton, well, now we've got that problem that we were just talking about. We've got that open space. There are a few new proteins here that we need to discuss. First, and we still have all of those same proteins, we're just not drawing them all. The first one that we have is this blue one on the left next to the calcium. This is a voltage-gated calcium channel. Voltage-gated calcium channel. It opens and closes in response to voltage, just like the voltage-gated sodium and potassium channels did. It opens at threshold, -55. Over on the right, there's another protein. It is a ligand-gated sodium channel. It could be different ligand-gated channels. It could be a ligand-gated potassium channel. It could be a ligand-gated chloride channel. We're just using ligand-gated sodium channel in our example. So the action potential is traveling where we just drew our red arrow over on the left. It gets to the terminal bouton, and the terminal bouton reaches threshold. What happens at threshold? Voltage-gated sodium channels open. But here, we also have voltage-gated calcium channels. So voltage-gated calcium channels open. Calcium high on the outside of the cell, low to non-existent on the inside, because calcium likes to bind to things, and we don't really want that. But now calcium is rushing in. And those voltage-gated calcium channels, they're all over this terminal bouton. So I'm going to draw another one down here. calcium rushes in. And what does calcium do? We keep saying it binds to things. One of the things that it will bind to are our little, let me see if I could draw this here, all of our little vesicles that contain our neurotransmitter, in this case acetylcholine. Now, when calcium binds to vesicles, it causes them to move. And remember how they're moving? We have all of those actin railways or microtubule railways around the cell. And they're going to move. They're going to move towards the edge of the cell. When a vesicle moves towards the edge of the cell and it comes into contact with the plasma membrane, what happens? Exocytosis, like we had in our not really attendance question. So all of those vesicles merge with the plasma membrane, exocytosing acetylcholine. So very quickly, the synaptic cleft fills up with acetylcholine. What happens with the acetylcholine? Well, acetylcholine is the ligand that this specific ligand-gated sodium channel responds to. So acetylcholine binds to the ligand-gated channel. What happens when something binds to a ligand-gated channel? It opens. This was, we said, a ligand-gated sodium channel. If it opens, what's going to happen? Sodium is going to diffuse in. As long as the acetylcholine is attached, that gate is open. Sodium rushes in. What kind of charge does sodium have? Positive. What's that going to do? It's going to cause the inside of the cell to become less negative, more positive compared to the outside. What's that going to do to the charge of the cell? Well, it's going to increase. It's going to depolarize. But the thing is, it's very, very local because we have the ligand-gated channels. In this case, they're letting sodium in. We have leaky sodium channels. We have leaky potassium channels. We have the sodium-potassium pump. But we do not have the voltage-gated channels on dendrites or on soma, cell bodies. So we are depolarizing, but it's very local. It's not going to propagate like an action potential does. This area that we're drawing is depolarizing. Areas away from this are not depolarizing. So we're going to take a look in just a moment to see what happens there. In this case, what we saw happen was the presynaptic neuron caused the postsynaptic neuron to depolarize. It brought it closer to threshold elsewhere in the cell. We will see how in just a little bit. But it made it more likely to send an action potential because it is bringing it in a depolarization direction. So when that happens, when the presynaptic neuron causes the postsynaptic neuron to depolarize and become more likely to send an action potential, we call that excitation. We are exciting the postsynaptic neuron. And that causes what we call a excitatory postsynaptic potential, abbreviated EPSP, excitatory postsynaptic potential. We caused the receiving neuron, the postsynaptic neuron, to depolarize. We caused it to be more likely to send an action potential. If we had a different neurotransmitter, if it was something other than acetylcholine, there are lots of them. If, let's say, it was serotonin or GABA or a few others, if it was a different neurotransmitter, it would have opened a different channel, a different ligand-gated channel. Maybe the channel caused potassium to leak out of the cell. Maybe the channel opened and allowed Cl- chloride to go into the cell. In either of those cases, we are causing the receiving cell to become more negative. We are hyperpolarizing it. In that case, we're making it less likely to send an action potential. We are inhibiting it instead of exciting it. In that case, we say that it is sending an inhibitory postsynaptic potential, IPSP, inhibitory postsynaptic potential. So that's how one neuron communicates with another neuron at a synapse when there is open space. Even though there were no proteins or membrane along that gap, we were still able to send a signal from one cell to another. So let's go back to our PowerPoint. Here is how your book presents what we just drew. Here is the presynaptic neuron at the top. That action potential that we drew in the last lecture is traveling down until we get to the terminal bouton. We have those voltage-gated calcium channels that open, allow calcium to rush in. Calcium binds to the vesicles that contain, in this case, acetylcholine. They merge with the plasma membrane, exocytosing the acetylcholine, which bind to our ligand-gated channels, opening and allowing sodium to rush in. So, we said that there are no voltage-gated channels on the dendrites or on the soma, so we don't get an action potential. Remember, action potentials in neurons always go from negative 70 millivolts up to positive 30 and back down. But regardless of the stimulus, it only ever goes as high as positive 30. Remember, action potentials are always the same magnitude. They always go to positive 30 and back. But we are not talking about action potentials here. We are talking about something called graded potentials. Graded potentials are what occur at a synapse, either on the dendrites or on the soma. In this case, a graded potential, it can have varying magnitudes. The greater the stimulus, the greater the magnitude. Maybe it only goes from negative 70 millivolts to negative 60. Maybe it goes to negative 5. Maybe it goes to positive 10 or positive 30 or positive 50. Graded potentials are dependent on the stimulus strength. And the other thing is we said that action potentials were very, very localized and they propagated. Graded potentials are localized, but the area that they affect depends on the degree of the stimulus. So if it were a very short-lived stimulus and it brought us from -70 as high as -40, it may only affect a very small area for a very brief period of time before it comes back down. But let's say it's a really strong stimulus and it takes us up to something like +15. Well, it's going to affect a much bigger area. And if the stimulus lasts for a long enough time, it will affect an even bigger area. So graded potentials depend on the stimulus, and that stimulus determines how high we go and how big of an area is covered. So, graded potentials could be very small, could be depolarizing, could be hyperpolarizing, could be very big, could be even bigger, could be even bigger. Same thing in the other direction. And additionally, they affect a very small area. They could affect an even bigger area. They could affect an even bigger area. They don't propagate, but the stimulus determines how big the depolarization or hyperpolarization is and how big of an area is affected. So we said different neurotransmitters. ACH was the one that we drew. Serotonin is another. There are many of them in your book. Don't worry about learning all of them. Just know that some of them are going to depolarize the cell and cause an EPSP. Some of them are going to hyperpolarize the cell, maybe cause an IPSP. There are different classes of synapse. Where is the presynaptic neuron communicating with the postsynaptic neuron? In this case, we have an axodendritic. The axon is connecting to the dendrite. This is what I said we were drawing. In some cases, the axosomatic, we have the axon of the sending neuron connecting to the soma of the receiving neuron. Axodendritic and axosomatic both behave exactly like what we just drew. But the other class is called an axonal synapse. In this case, the axon of the sending neuron connects with either the axon hillock or the axon of the receiving neuron. We're going to see why that's important in just a moment, but I'm going to click on this. I don't know if there's going to be any audio, but you can click on it in the PowerPoint that's loaded to watch it if there is no audio.
Our brains are constantly processing information, which requires communication between billions of nerve cells. Signals are passed from a sending neuron to a receiving neuron at a junction called a synapse. An action potential in the sending neuron travels down the axon until it reaches a synaptic terminal. The narrow gap between the synaptic terminal and the receiving neuron is called the synaptic cleft. The synaptic terminal of a sending neuron contains numerous vesicles filled with neurotransmitters, chemicals that carry information across the synaptic cleft. When an action potential reaches the synaptic terminal, the vesicles fuse with the plasma membrane of the sending neuron, releasing neurotransmitters into the synaptic cleft. The neurotransmitters affect the receiving neuron, changing the distribution of charge across its membrane. Let's take a closer look. An action potential is propagated down an axon by the opening and closing of sodium and potassium channels. When an action potential arrives at the synaptic terminal, it causes the opening of calcium channels, shown in green. Calcium ions enter the synaptic terminal through the calcium channels. Calcium ions bind to the vesicles containing neurotransmitters. This causes the vesicles to fuse with the plasma membrane of the sending neuron, releasing neurotransmitters into the synaptic cleft. The neurotransmitters diffuse across the synaptic cleft and bind to receptors in the plasma membrane of the receiving neuron. Here, the receptors are ion channels that open. Ions move across the membrane, changing the distribution of charge across the membrane. The neurotransmitters are quickly removed from the synaptic cleft, ending their effect on the receiving neuron. A single neuron can...
So here we're going to look at what happens if multiple action potentials are sent to a single neuron. There's different effects that can happen. So that's what this video is going to demonstrate now.
Receive signals from many sending neurons. The signals arriving from the axon on the left are excitatory. They make the receiving neuron more likely to generate an action potential, as indicated by the green glows. Signals from the axon on the right are inhibitory. They make the receiving neuron less likely to generate an action potential, as indicated by the red glows. The two sets of signals cancel each other out, and no action potential is generated. Now, only excitatory signals are transmitted, and the receiving neuron generates an action potential down its axon. The signals that pass between these and countless other neurons constitute our thoughts and coordinate our activities.
So we could have many neurons communicating with a single neuron, and those signals that are sent will stack with each other. Let's take a quick break and do a thinking cap question before we carry on with that. Pinched nerves can result in either numbness or difficulty in moving a limb. Why would that be? This relates to our questions that we had at the start of the lecture, are not really attendance questions. This one can be a real thinker. So go ahead and pause the video and try to see if you can come up with the answer. But don't feel bad if you can't, because it really requires thinking deep about everything that we've talked about so far and putting it together. So if you're wanting to move, or if you are feeling a sensation, remember, all that are electrical signals, action potentials. And those action potentials are typically passed through a few neurons along the way. Now, as we just saw, one neuron will communicate with another neuron using neurotransmitters. What are neurotransmitters? Generally, they're proteins. Proteins that are made where? The rough endoplasmic reticulum. Where's the rough endoplasmic reticulum? In a neuron. This was back in our very first lecture of this chapter. Up in the soma. So here is our neuron. We have the proteins made here in the soma, those neurotransmitters. They're packaged in vesicles. And then those vesicles are stored where? down here in the axon terminal boutons. Well, if you pinch a nerve, let's say we pinch a nerve right here, what's going to happen? Eventually, we're going to exhaust all of our neurotransmitters that are stored down in the axon terminals. And more will try to travel down the axon, but they can't. They're blocked off. So at some point, this neuron will not be able to communicate with the receiving neuron. Action potential cannot be passed, signal cannot be passed, then either you cannot feel sensation if it's a sensory neuron, or you cannot move muscles if it's a motor neuron. So what about reuptake of the signal, or a termination of the signal? How do you stop that action potential from traveling from the first neuron to the second? We have to get rid of those different neurotransmitters. Otherwise, they're going to stay bound. So what happens? One option is reuptake. The sending neuron is just going to pump that neurotransmitter back into itself. That will cause the neurotransmitter to no longer be in the synaptic cleft. Sometimes this happens a little bit too quickly. We have reuptake happening before the signal can be communicated. One common thing that most students have heard of is a class of drug called SSRI. That stands for selective serotonin reuptake inhibitor. So in the case of serotonin, sometimes it is going to go through this reuptake process quicker than it needs to. It doesn't stay in the synapse long enough to do what it needs to do. The sending neuron pumps it back in too quickly. Selective serotonin reuptake inhibitors, SSRIs, selectively inhibit that reuptake of serotonin. They allow it to stay in the synaptic cleft longer so that it can communicate. Another option is degradation of the neurotransmitter. This is done by enzymes. One enzyme that breaks down acetylcholine, for example, is acetylcholine esterase. It takes ACH and basically just breaks it in half. Another option is just diffusion. If the sending neuron stops exocytosing its neurotransmitter, eventually we're not going to have a concentration gradient. And as we are pumping some of that back in, well, the concentration gradient will go in the other direction. So it will diffuse away from those ligand-gated channels. In all of those instances, and really all three of them are happening at the same time, the result is we stop the signal being sent. Everything we've talked about so far are chemical synapses, but there is something called an electrical synapse. In an electrical synapse, two axons are joined together with gap junctions. And remember, gap junctions allow substances, in this case ions, to move back and forth between two cells. So if we have an electrical signal traveling through one cell, those ions can pass through gap junctions into the neighboring cell. We've already talked about EPSPs and IPSPs. And in our video, we saw, without using the word, summation. We saw what happens when more than one signal is sent to a receiving neuron. Well, there's different ways that we can have summation. We can have temporal summation. One neuron is sending signals to the receiving neuron very, very quickly. Just one after another, after another, after another. In that case, we get temporal summation. We get a little bit of a stimulus, in this case excitatory, but it could be inhibitory. But if another signal comes through before we get all the way back to rest, they will start to stack. And we only have to get to negative 55 to cause an action potential. If it is in a dendrite or a soma, then it will affect a bigger area and have bigger waves. Spatial is what we saw in the video. It is when more than one neuron is sending signals to the receiving neuron. In this case, they will stack just because they are sending signals at the same time. Temporal, one neuron sending signals quickly. Spatial, multiple neurons sending signals. And the result here is kind of what I was just drawing. If we have two excitatory signals that come through after we get back to rest, then there is no summation. But as I was trying to draw with my wave earlier, if we have another signal come through before we get back to rest, as we see right here, then we get summation. Spatial, if two signals come through, or more than two, more likely, then again, we get summation. But sometimes signals are in the opposite direction, like we saw in one of our animations. Maybe we have an inhibitory signal, makes it less likely to send us an action potential, and an excitatory signal at the same time. In that case, we don't get an action potential. They cancel each other out. Before we get to the different types of circuits, I want to go back to our example right here. We said that axosomatic and axodendritic are what we drew. We said that we drew an axodendritic, but we also said that it would be exactly the same if it was an axosomatic. Because we get graded potentials, and we get graded potentials because why? Because there were no voltage-gated channels. So we get a signal, and then maybe we get a bigger signal, maybe we get a bigger signal based on the stimulus. But I did not say that it was the same as an axo-axonal. And that's because right here, what is this guy? The axon hillock. In all three of our lectures, we have said that the job of the axon hillock is to determine whether or not an action potential will be sent. How might that happen? Well, if we get a big enough graded potential that it causes the axon hillock to reach threshold, an action potential is generated and it travels the length of the axon. Does that make it any more clear as to why or how the axon hillock generates an action potential or doesn't generate an action potential? Because up here in the soma, in the dendrites, if we reach threshold, nothing special happens. It is only if the axon hillock reaches threshold. Because the axon hillock does have voltage-gated sodium channels. What was the job of the voltage-gated sodium channel? He was the guy who kicked off the action potential. He started the first domino falling. So, soma, dendrites, they do not have action potentials. They do not have voltage-gated channels. Axon hillock does have voltage-gated channels, and that's why he is able to generate an action potential. And once we generate an action potential, it is propagated. There is no stopping it, and it will travel the length of the axon. That is how the axon hillock determines whether an action potential will be generated and passed, because the axon hillock is the first spot to have those voltage-gated sodium channels. Let's go back and use another thinking cap question here. It's not on our show here, but let's go ahead and use a thinking cap question based on this. Which of these three types is most likely to result in an action potential? Is it an axosomatic, an axodendritic, or an axo-axonal synapse? Which of them is most likely to result in an action potential in the receiving neuron? Well, axo-axonal would be most likely, because axo-axonal only has to get to -55 in a very small area. Axosomatic and axodendritic will have to have much more stimulation to get the axon hillock to a point of threshold. So here are some different types of circuits, how one neuron can send a signal. The first one is called a diverging circuit. We have a single neuron that sends a signal to a few more neurons. Each of those new neurons generate an action potential and send it along to a few more neurons each, generating a new action potential, sending it along to a few more neurons each. The result is one single neuron can cause action potentials in many neurons. So, for example, you could have one single neuron in your brain stimulating a lot of motor neurons just by sending a single action potential. Next is kind of the opposite. It's called a converging circuit. We have lots of sending neurons, but only a single output neuron. Sometimes you'll see this called a concentrating circuit. And in this case, maybe you have information coming in from your eyes. Maybe you have information coming in from your ears. Maybe you have information coming in from touch, whatever. All these different sensations can invoke the same memory. Because when you had the memory first built, you were seeing something and at the same time hearing something and at the same time maybe feeling something. And they all kind of built that same memory. Next, we have a reverberating circuit. A reverberating circuit, one neuron sends a signal to a second neuron, who sends a signal to a third neuron, who sends a signal on and on and on, but then it feeds back to a neuron going the other direction, who sends a signal to a neuron somewhere along the pathway. The result is the first neuron sent a single action potential, but then it just keeps going. It keeps going, even though that first neuron is not sending an action potential anymore. He started it, but it's kind of self-propagating at that point. This is why you do not have to think about breathing. It's kind of automated. It just kind of keeps going on its own repetitively. And lastly, we have something called a parallel after discharge circuit. It's kind of a combination of the first two. One neuron sends a signal to multiple neurons, but then those multiple neurons all send a signal back to a single neuron at the end. And it says that this is kind of for mathematical calculations, but there is really good evidence that this type, the parallel after discharge, is kind of responsible for the sense of déjà vu. And that is if one neuron sends a signal and it gets to another neuron. But let's say there are more neurons in this pathway. So the same signal gets there, but it takes just a fraction of a second longer. You get the same stimulus at two different points in time. That first stimulus, and then right after that, you get the same thing, and you think, it seems like I've felt this before, but really it was just a moment before. It may feel like it was days or weeks or months or years before, but it was really just a moment before. So there is really good evidence that parallel after discharge circuits are responsible for deja vu. Let's wrap up with the thinking cap question. If you've ever walked into a place and smelled something from your childhood, you typically have instant, vivid memories. If you see photos of that same time, you will also have those same memories. How can seeing something and smelling something have such a similar response? Let's look back at our different types of circuits. Remember, maybe we saw something. Maybe we heard something. Maybe we felt. Or maybe there was even another one. Maybe there was one where we smelled something. All of those can invoke the same memory because they are a converging circuit. Multiple different things built that memory, so multiple different things can trigger that memory. All right. It says next time exam 3. That is not true. Actually, next time we have the muscular system. So we've already had exam 3. You can ignore this. Next time is the muscular system. All right. I will see you all in the next lecture. Make sure to ask questions. We are in some pretty challenging material right now. This is not the time to put those questions out of your head. Make sure to ask them so that I can help. I hope you enjoyed the lecture. I hope you enjoyed this chapter. Keep that action potential stuff fresh in your mind because we are going to talk about it again in our next chapter. All right? Take care, everyone.